Thank you for your patience. Good afternoon. My name is Clint, and I'll be talking about how Xenon Pharmaceuticals uses automated electrophysiology to accelerate its drug discovery efforts. Specifically, Xenon Pharmaceuticals is actually interested in looking at ion channel modulators, and in particular, blockers of voltage-gated sodium channels. And we're looking at molecularly selective blockers for NaV1.7 ostensibly to treat pain, mainly because this is a very well-validated target. Specifically, a lot of people have worked on it. In fact, Paul Goldberg from Xenon was one of the first to actually document back in 2007 in clinical genetics that mutations, loss-of-function mutations of SCN9A, the gene that encodes for this particular sodium channel, results in patients having loss of pain, congenital indifference to pain. And further research by Waxman's group and Pfizer and everybody else has implicated NaV1.7 in being essential for the pain pathway. And so there's a lot of effort looking at blockers of NaV1.7 that are molecularly selective. And in fact, Xenon has been working in a collaboration with Genentech to look for such molecularly selective compounds to move forward in clinic to treat pain. At the same time, Xenon has a specific in-house project looking at molecularly selective blockers of NaV1.6 to treat severe childhood epilepsy, for example, Dravet disease. That being said, our EP team is really responsible for generating and helping the MedChem effort to look at structures and also optimizing leads to find such compounds that are really molecularly selective and at the same time very potent. And so with Xenon, we've been looking at automated electrophysiology to accelerate our drug discovery process. And our experience is such that when I joined in 2004, there were basically only 4 electrophysiologists, and they're all doing manual electrophysiology at the rig. And it was very hard to generate data. So within 2 years, we purchased the Patch Express, which at the time was quite revolutionary. It had 16 channel recordings. Later on, we upgraded to the Q-Patch 48. So within a span of a few years, we went from 4 people doing manual to quadrupling our efforts with the Patch Express, and then later on quadrupling again with the Q-Patch 48. And more recently, we purchased the Cube 384, which is a high throughput system, which compared to the 48 channel has now, it's not quadrupling, it's sort of octupling. So it's eightfold the output. So essentially, you could do 384 recordings. So I'll talk a little bit about how we use the Cube in our efforts. So here's a picture of the Cube here in black. Ostensibly, we use it to really expedite our drug discovery. And you can actually use the Cube to, other people use it, can use it to basically do high throughput screening of their libraries, or even safety pharmacology screening, for example, NaV1.5 or even HERG. But we do something a bit different at Xenon. We don't actually use it as this primary assay. We actually have other assays that do the upfront heavy lifting. We actually use the Cube as a complementary system to help confirm and enrich our HTS assays. In particular, I mentioned we have a pain assay and an epilepsy assay. For those 2 particular projects, we have a ligand binding assay for one of the projects and a fluorescent-based influx assay for the other project. Those do the heavy lifting. And what we do with the Cube is actually confirm the results for the selectivity, for the potency. And what we find is that it's between 3 to 10-fold matching the types of potency we find for those assays. At the same time, those types of assays can actually, in some cases, look at molecular selectivity, and we confirm it as well with the Cube. And so it actually enriches those findings, and we move forward. And the reason why we do that is because each particular assay, whether it's ligand binding, flux, or electrophysiology with the Cube, have different strengths and weaknesses. And we can talk a little bit more later about that if you want, but just as a summary, the ligand binding assay can actually have steady state values because you can incubate your compounds overnight for 24 hours. That's good. But it is a binding assay and not a functional assay. The fluorescent influx assays can be a shorter time, but it actually uses exotic toxins to elicit signals. So that could be a modification of the channel. The advantage of the Cube is it could be a 3 to 4 system, and people think of it as the gold standard. However, the challenge is that you might not reach the steady state that you're looking for, specifically for really potent compounds, which require a long time to reach steady state. So that being said, we try to make sure that we aren't misleading ourselves with each particular platform, and we try to get all the platforms to agree and then move forward. That being said, the cube is also not only used to confirm and enrich our HTS assays, but also to understand mechanism. And in particular, we have been able to look at the mechanism of action of our particular molecularly selective NaV1.7 blockers. And in collaboration with Genentech, we published that finding in Science, and I'll talk a little bit about what we found. So just for the, so the general background, talking about NaV1.7s for the uninitiated, I'll walk you through the NaV1.7 followed by how we use the cube for enriching data, followed by mechanism of action. So in terms of background, the NaV channels are, you can think of them as being a family from NaV1.1 subtypes all the way to NaV1.9, and they're found in different populations across the body. And we can think of it as a predominance of NaV1.1, 1.2, 1.6 in the brain and spinal cord. In the skeletal muscle, it's generally predominantly NaV1.4. Cardiac muscle, as you know, is NaV1.5. And I'll focus on the DRG because we're looking at pain. In general, NaV1.7, 1.8, and 1.9 are here in the adult tissue, and 1.3 is only there when you have injury. Now, that being said, what happens when we're talking about safety pharmacology when you actually block these channels in a normal patient? Well, here, what you'll find is that in the CNS, if you actually block 1.1 or 2 in a normal patient, it can lead to proconvulsive issues. You can cause convulsions. NaV1.6 actually is also found exquisitely in the nodes of Ranvier. And what that means is if you block that, you can get nerve conduction problems, very similar to like MS. So you don't want that. At the same time, if you block 1.4, you would get muscle weakness. You block 1.5, you get arrhythmia. And if you block 1.7, 1.8, or 1.3, if it's only exclusive in DRG, you would think that actually it would have just no pain, just be it analgesia as a side effect. Given that our projects are focusing on 1.6 and 1.7, we don't want to block any of the other ones because they would lead to safety issues. Now, that being said, let's back up. So with those channels, what we do in-house is that from NaV1.1 all the way to 1.9, we've been able to look at 8 NaV isoforms from 1.1 all the way to 1.8. 1.9 has been extremely difficult to create in-house, as everybody knows, except probably at Icogen and others. That being said, we also have non-human orthologs of our NaVs, and this is the type of consumable that's used on the cube. It's actually a 384-well chip. It's called the Q-chip. And in the bottom of each well is either a single hole, so you capture 1 cell per well, or it's a multi-hole. And we focus on 1 cell per hole. And the rationale for that is we find it actually gives better recordings and more stable. I'll talk about a little bit reasons why later if you want to ask that question. And in our electrophysiology experiments, we actually run it for an hour. I know that for some other industry pharmaceutical companies, they actually run HTS assays that are really short, in the 15 to 20 minutes. And we're really moving it forward to 1 hour. It's quite an extreme. And we are actually well known to push all our technologies to extreme. And I'll talk to you about why we think 1 hour is appropriate and how we make sure that we aren't having too much rundown or artifacts in our data. That being said, we're able to generate 10 8-point IC50s per plate. And in general, within a day, we could run anywhere from 4 to 5 plates on a routine day. What that translates to is 40 to 50 IC50s. Now, when we have these particular cell lines, how would you start from the ground up to make a sodium channel project? Well, first, you have to have your cell lines. And how do you do that? Well, you have to have your molecular biologist to create the cell lines for you. But then you have to weed out the best cell lines. So how do you do that? Well, as it turns out, let me back up a little bit. This plate is actually 16 rows by 24 columns. And the way the cube works is it can actually deliver the cells across in rows. So you can actually have 16 cell lines that can be recorded at any one time. And so Sophion makes different types of these cell transfer plates, anywhere from a single lane, so you're only looking at one cell line, or in this case, 16 lanes, so you can look at 16 cell lines at once. What I'm going to talk to you about is we're going to look at one where we've looked at 8 lanes or 8 cell lines all at once in the single experiment. This is just a proof of concept to show that if you had to look at a particular cell line, how do you define or find the best cell line that the molecular biologist created for you? In this case, let's hypothetically choose this as a NaV1.7. The molecular biases have created 8 different cell lines, and 384 divided by 8 means you have 48 cells per well. So what we're going to do is we're going to look at each of the cell lines and look at its basically its current density. And the way we measure success is the success rate, how many cells live at the end of this particular experiment, the expression levels in terms of its average current density, as well as uniformity. So as an example here, what we'll look at is number 7, cell number 7. It's basically quite uniform, but in general, it's below what the other cell lines are doing. So we wouldn't choose that cell line. If you look at cell line number 2, what you notice is that it's incredibly uniform, but that's only because there's 4 cells. Its success rate is horrible. It's only 8%, 4 cells out of 48. So which ones would you choose here? Well, you might choose number 5. Unfortunately, although it's got the same sort of height or current density average, it too is poor in terms of success. In fact, we would choose number 6 because in general, you'd get a majority of cells that are 52% success. You'd have 1, 2, 3, 4, 5, maybe 6 cells that are dropping below the average, but in general, this is the one you would choose. This is just to show that you can choose a cell line just based on one experiment, because you have enough data to drive it forward. You could keep running replicates, but basically you can actually quickly choose the cell line you want to have. That's one of the ways we were able to optimize our cell lines and move it forward. Now, notice that this is picoamps or picofarad. The average of this is anywhere from 7 to 8,000 picoamps per picofarad. Given that the average cell size is about 10 picofarads, that's 7 to 8 nanoamps. Now, that's a big current. And we get that substantially, not just for 1.7, but for the majority of our cell lines. Now, with that said, what we're going to do is we're going to actually look at some biophysics to characterize these cell lines. And what you'll notice is that this is an actual snapshot of the traces taken from the cube, where we're doing a really basic 100 millisecond IV. Here's the activation, and here's the inactivation, holding at negative 140. What you'll notice is that the currents are actually upward. And that's because we use flipped gradients. So because our currents are so big, generally 7 to 8 nanoamps, 1.5 we found can exceed 10 nanoamps easily. We found that flipping the sodium gradients allows us greater voltage control. And at the same time, serendipitously, we found that in flipping the gradient, we were able to have greater stability in our recordings. So instead of just recording for a mere 20 minutes, we could extend it out to, in this case, 70 minutes or more. Now, with that said, that's only for some cell lines, but in general, flipping the currents has been really beneficial for the cube. Now, with that in hand, what we're going to do is use those kind of biophysics where I mentioned before, you can run 16 cell lines or 8 cell lines at once. Here, I'm going to look at 4 cell lines at once using the IV. And 100 millisecond, just like before. And I'm going to look at these 4 NAV subtypes. 1.5 for cardio safety, 1.7 as our pain target, 1.6 as our epilepsy target, and 1.7 as our off-target proconvulsant channel, which we don't want to interact with. But the reason why I mention this is that this particular experiment was run in one experiment. And we can generate steady-state inactivation curves quite quickly with this. And so just basically, you can see that 1.5 is left shifted, it's inactivation, and then followed by 1.7, 1.6, and 1.1. So really what this allows you to do is very quickly understand the types of performance of your cell lines with the cube, with the types of composition of your internal, external solutions that you have, and design your voltage protocols so that you can have a good way to assess the block of compounds systematically for all these subtypes. Notice that the success rates are 36, 49, 63, and 29. This is all in flipped sodium. So it's saying that 1.6, 1.7, in this case, in general, that's a low number. Typically, we get more than 50%, hovering about 60% for all these cell lines. This is just an early example. So knowing the biophysics, we're going to design our voltage protocol to look at pharmacology. So this is the cube using biophysics. So moving to pharmacology, what we're going to do is we're going to now, as I mentioned before, we use the cube extensively to confirm and enrich our already pre-existing high-throughput systems, whether it's ligand binding or flux. So what we'll do is we're going to, in this case for NaV1.7, we're going to use this particular protocol. We're holding at -60 because what we found over here is that if you hold it at -60, you're basically going to inactivate majority of your channels. And it'll be in the inactivated state. So we do a test pulse to ensure that it's completely inactivated. We recover with a 20 millisecond repolarization to -150, which will quickly bring things out of inactivation. And hopefully that's short enough to not allow any compounds to unbind. And we give a test pulse every second. And so what we do is that we have a vehicle that's been running for 40 minutes, and then we add a compound. And in this case, we have vehicle, which you can show. You'll notice here it's like 4 additions. There's actually 4 additions here. And so that's why you see this sort of artifactual dip. That being said, you can have a vehicle rundown. And what you can do is, with this particular protocol, with this highly potent compound, which has an IC50 of 0.46 nanomolar, at 1.8 nanomolar, it actually takes a long time to block, up to 10, 20, 30 minutes to achieve steady state. Now, I want to point out 2 things. Typically, when you run a HTS, when you run either a library screen or a safety pharmacology, you're usually dealing with micromolar compounds at single concentrations. For if you're screening a library of 100,000, you're going to test them all at 5 or 10 micromolar, 1 concentration. The advantage of that is that you're going to have fast kinetics. Your block is going to be really quick. And specifically for safety, you already know that it shouldn't be hitting that target, so you already have it at high concentration anyways. The reason why I mention it is because this is a sub-nanomolar compound. It will take a long time for this to reach equilibrium. So this is, even at 3 times the IC50, or 3 times 1.8 nanomolar, 5.6, it's going to take 20 minutes. So you can imagine getting this sort of data will take a long time. Now, the reason why I mention it is because we are able to achieve stability, achieve steady state for sub-nanomolar compounds, and record well over 60 minutes. And the fact that we record vehicle is very important in this context. We are able to generate, in this case, 10, as I mentioned, 10 IC50s, 8 point on a single run. But we also record the vehicle rundown. So what we have here, 8 point IC50s for 10 different compounds. And because we have the vehicle rundown, we can correctly correct for the rundown. That, for example, here's a blow up. So typically, if you get an IC50 that's 8 point, 1, 2, 3, 4, 5, 6, 7, 8, around 8 point, you notice here the IC50 is not at 0.5. This is extrapolated in line a little bit higher. And the reason for that is that this experiment had a 0.08 or 8% rundown. So if you didn't have this, you would think that this was more potent than otherwise. Okay? So we're going to correct this by shifting the IC50, or shifting the normalized inhibition to a point where it takes into account the vehicle rundown. That makes sure that you actually have more accurate IC50, something that we weren't able to do even on the Q patch 48. So we have rundown correction for all our IC50s now. Okay, so in validating the system, what we'll do then is we'll get reference compounds and compare that to manual electrophysiology data in literature for those very same compounds. In this case, things like carbamazepine, lamotrigine, lidocaine, ranolazine, all these against literature values. And what we find is that this particular protocol allows us to get fairly good agreement with literature values. And so that's very reassuring. That's for that potency for 1.7. So what we'll do then is we're going to look at all the other subtypes. In this case, we'll use similar protocols appropriate for that particular subtype and generate IC50s. In this case, for tetracaine, which is known to be non-molecularly selective, with our protocols, we show it's non-molecularly selective for all the subtypes from 1.1 all the way to 1.8. So with that in mind, we know that our protocols are aligned with literature. We know that our subtype selectivity is as we expect because non-selective are non-selective. And then now we can actually use our data from our cube to see what it looks like compared to our ligand binding assay and our flux assay, which we show here. So our cube IC50 is on the bottom, ligand binding assays on the y-axis, and it's actually in fairly good agreement between 3 to 10-fold. So for our primary assay for one of the projects, it's basically 3 to 10-fold, not too bad. For a non-functional assay, this is a binding assay. At the same time, when we look at an influx assay, we show there's a 10-fold shift, but in general, it is ranking properly. So these are going to do the heavy lifting for those particular projects that we are interested in, and then we'll follow up with the cube to take a look at those compounds of interest that either are very potent or meet tentative molecular selectivity that we're interested in. Now, with that said, we also use the cube for exploring biophysics and mechanism of action. So, as I mentioned before, in our collaboration with Genentech, we published back in December last year the structural basis of a molecular selective NaV1.7 inhibitor, and we published this in Science. And in this collaboration, we were actually able to define the binding sites and use the automated electrophysiology, the cube, to generate the molecular selectivity as well as to elucidate a little bit more about the mechanism. So let me walk you through what we did. So using the protocols, as I mentioned before, intetracrine was non-selective. With these, with this particular compound, GX674, we demonstrate that it is very much molecular selective, although it is 1.7 and 1.2 basically equal, followed by 1.6, and it doesn't hit the other channels. Of interest here is, as I mentioned before, because this is a safety pharmacology talk, well, this would be good for hitting 1.7, okay, pain, maybe 1.6, okay, great, it'll maybe treat epilepsy, but it hits 1.2, maybe it's proconvulsant, not good. That being said, what we want to do is we actually want to find out how this compound binds to the sodium NaV1.7 channel. And that's what we did in collaboration with Genentech. So how do we go about doing that? Well, typically what you want to do is you really want to get a crystal structure of the native NaV1.7 channel, but that's been incredibly difficult. And you can't get crystals, but as it turns out, you can actually get ligand binding on the native channel. So what can you do? What can you get crystals from? Well, Gian Piande, who worked in basically in 2011 in Nature, published in Bill Catterall's lab, he basically was able to find that this bacterial channel, NAV-AB, could get crystals. So he joined Genentech, and we worked with him, and basically found that, hey, great, we can get crystals. But when we try to put a GX compound on it, nothing bound, unfortunately. So the next step was, okay, let's try to find something that can get crystals and bind. So they thought, well, there was really tentative experiments pointing to region 4, domain 4, as being important for the ligand, for the binding. And so what he did was, he basically did a chimera and put in VSD4 and merged that with the NAV. So the pore module where the ion conducting is right there, and here's the voltage sensing domain. So basically, he switched out the voltage sensing domain, but kept the the gating here for the NAV-AB. So it's a chimera. Unfortunately, there was no crystals, but provocatively, there was ligand binding. So it's like, it's binding, but you can't get crystals. Well, that's almost like that. I mean, it's like no crystals, ligand binding, but it's binding. What can we do? So what Gian did, along with Chris Koff, was he had this brilliant idea. Okay, well, let's try this. Let's cut this thing in half and just have the extracellular domain sitting in there. And it's a chimera, but it's like a chopped off chimera with only the extracellular component exposed. And what he found, lo and behold, was you got ligand binding and you got crystals. So with that, they crystallized it. But then the question was, well, you've got this native NaV1.7, but now you've got this chimera. How real is this in terms of its ability to mimic this? Because this is a homotetramer. It's taking this and multiplying by 4 to recapitulate the sodium channel as a chimera. So how can you determine whether these are equivalent? So what they can do is actually test 20 compounds in our series and look at the ligand binding here and compare it to the ligand binding there and see if they're basically equivalent. But the reason why we can't do this with electrophysiology, unfortunately, is that while we can record in native channels, this chimera, there are no conductants that we can find with electrophysiology. So using ligand binding, they did this comparison of, well, here's the native channel ligand binding values compared to the chimera. In general, there's a little bit offset, but they actually track. And as we mentioned before, the ligand, the functional assay with electrophysiology tracks quite well with the native channel by itself. So in general, the thinking was, well, it seems to perform similarly enough to the native channel to say that, yes, it's functionally similar. Okay. That being said, it's pretty provocative in that, well, if this is on the extracellular side, we can actually determine with electrophysiology whether that's true. In other words, we want to confirm whether the binding site is extracellular. So what we can do is we can actually, what experiments can you do to determine if this is extracellular? Well, one way is to add compound extracellularly, but look at the on rates for a neutral version or a charged version. And the thinking would be that if the binding site is on the inside, the charged version would be slower to get in because it's charged. So we did that experiment. But before doing so, we looked at the potency, and what we found was, here's the neutral version, GX395, here's the charged version, GX626. And what you find is the IC50s are a little bit shifted. It's actually a little bit more potent for the neutral and less potent for the charged, which sort of indicates, well, maybe it's slower to get in. Now, that being said, that's similar enough, but what we can do is actually look at on rates of compound. So here we have it here. This is actually a graph, an IT plot of multiples of the IC50. So in this case, it's 3 times IC50 followed by 5, followed by 8, followed by 16 for the neutral and for the charged. And in general, they seem to be matching in terms of their profile. If you look at the taus themselves, what you find is the absolute numbers are sort of left shifted for the GS626. But if you normalize it to the folder of the IC50, they're basically overlapping. So when you apply this extracellularly, they seem to have similar on rates when you normalize the folder of the IC50. It suggests that maybe it is extracellular, but you can't discount that it still might be intracellular. This is not a definitive experiment. So what is the definitive experiment? Well, applied intracellularly, and that's what we did. On the cube, you have the ability to change your internal solutions. And so we actually started off with putting compound in the intracellular solution. So the hypothesis would be, if you put it inside, will it block? Very simple experiment. Before doing that, we want to use a reference to make sure that our system is working properly. So to do that, we put in QX314, which is basically the charged version of lidocaine. And in intracellular solution, just nominal, basically there's no, it's green, basically you have this current density. And as you increase the concentration of QX314, you get less, basically more and more block. So this is a positive control. It's basically 52 micromolar. That's what we expect. So definitely it works. So what happens when you put the GX compound? Well, if you put GX262, the charged version, at 50-fold the IC50, it does nothing. So this is pretty convincing that in fact, the molecular selective site is not accessible from the intracellular side. It must be on, it's probably the outside. Now, what I've done then, I just wanted to highlight what Xenon's doing with the cube or automated electrophysiology for drug discovery. We actually have been able to expedite our electrophysiology efforts. We don't use it really as a primary screen or safety per se. What we do is actually we look at channel biophysics to optimize protocols to generate protocols appropriate for molecular selectivity and actually look at mechanism of action. And you can design your protocols under voltage clamp and or current clamp now, and with temperature control. And so what I've demonstrated in this presentation is looking at steady state inhibition at different voltages for different subtypes, as well as kinetic analysis of on rates, but you could also do off rates as well. And I haven't shown it here, but you could also look at pulse frequency, frequency dependence. And we've also tried in relation to the Novartis' talk by Laszlo, we've tried iPSCs as well. Very hard to work with. The success rate of here is like 0.1%. We have one cell. So it's very difficult to work with iPSCs, but I think that as we develop more mature and more experience with iPSCs, that's the hope, is actually put this on this machine. Because having a patient population with that particular mutation and seeing how our compounds interact with that is something that we're keenly interested in. So that's the end of my talk. If you have any questions, please feel free. Thanks.
That was very interesting. Also brought back memories of when I was running sodium channels on the Q patch.
Yeah.
We know, so you're control rundown, we found that that was generally due to shifts in activation of the V half. Would you have the same insight?
So we looked at the V half as well over time. It does shift. It actually sort of left shifts over time. But I think, what we do at the very end is actually look at the compound blocking at that time point isochronally. So everything is isochronal to the vehicle. So what you can do then, and that's why we're confident that we can actually adjust for that vehicle rundown or shift and make our ICVs more adjusted for any apparent rundown or shifts in your VHAVs.
Right, we were running cumulative IC50s, so that shift affects your potency, especially when you're looking at state-dependent compounds. And my recollection is your compounds are very state-dependent.
Very state-dependent. So we've actually run the protocols across different voltages, and we see it basically going from sub-nanomolar, boom, okay, all the way up to hundreds of nanomolar when you go to very negative, as you would expect. And with our protocols, you can actually look at the steepness of that particular voltage dependence, which is kind of interesting. So in other words, you can go -60, -90, -120, -150, and then look at the IC50s and look at the steepness of that voltage dependence, which is interesting to us because when we look at physiological resting membrane potentials, we want to make sure that, okay, is it so voltage dependent that all of a sudden when you're at -90, now it's almost resting state block, So we keep our eye on those voltage-dependent shifts in IC50s.
Have you looked at the role of beta subunits in setting that voltage dependence?
That's a great question. Yeah, for us, we focus exclusively on beta 1, but for some subtypes, we looked at beta 1 and beta 2. We did some exploratory experiments with beta 3. We haven't looked at beta 4 per se, but yeah, I mean that's something that with this machine, we have the ability to look at all the different permutations of different betas as well.